you know. All right, so what do we do when we have webinars like this? During the webinar, guests usually bring together experts from different parts of the world to discuss various topics on global health issues, and then to also come up with recommendations for policymakers and also to enlighten the public on what is going on in this area. It's so important to also let us know that the chat box is open. I know that only our panelists would be able to share their videos and also use their audio, because the platform we're currently using is the Zoom webinar. And but our audience, our participants can actually make use of the chat box to drop information, to introduce themselves, to introduce where they are joining from. And also, we also have the question and the answer box, where our participants can also drop their questions and their their questions for our participants. So having established that fact that participants can use the chat box to drop, to introduce themselves, and everybody's going to see it, they can also use the question and the answer box to also drop their question. I will be officially starting the webinar, and then I will be starting with introducing my organization. All right, so the Global Emerging Pathogens Treatment Consortium, GET, was established in 2014 as a direct response to the 2014-2016 Ebola virus disease outbreak in West Africa. And also, I'm also glad to let you know that GET now operates firmly in the African biosecurity space, and we function as a think tank where we provide high-level advocacy, research, and operational and necessary expertise to support countries and communities to combat emerging biosecurity threats. I will also like to let you know that GET activities are quite numerous, and then GET is registered in Nigeria, GET is registered in Ghana, and GET is registered in the United States of America. And all of our projects can be classified into 5. GET is involved in capacity building programs. GET is also involved in research, and GET is also involved in policies, publications of policy brief, newsletters, technical papers. GET is also involved in logistics, and GET is also involved in policy advocacy. GET is involved in outreach and community engagement. If you want to know so much about our organization, I would like to also implore you to go right on our website, maybe after this webinar, at www.getafrica.org. I have our communication team on board to drop all of this information in the chat box for us to view. So let's go right into today's webinar. The topic for today's webinar is understanding the benefits and also the potential risk of synthetic biology. I'm going to say that again. Today, we're going to be focusing on understanding the benefits and the potential risk of synthetic biology. And then I would like to give a background to this, to what we're going to be talking about this before I introduce our speakers. So right here, what I have here is synthetic biology has been globally recognized as a driving force for the concept of bioeconomy. It has been used to generate diagnostic tools that has improved the care of patients with infectious diseases. Through synthetic biology, we've been able to produce vaccines. We've also been able to develop organisms that can synthesize essential chemicals such as precursors for antibiotics and polymers. Synthetic biology is one of, I would call it an innovation of the modern biology, making biology more useful in diverse ways. But today, it is very important that what our speakers are going to be doing for us today is that they're going to be making us understand in details the concept of synthetic biology, and especially why African countries should embrace the concept of synthetic biology. Why today we're also going to be focusing on understanding the benefits, the amazing benefit that can come out of synthetic biology, while not forgetting the fact that there are potential risks. If science is not handled, science can become a threat. There are potential risks that we also need to understand. Okay, so it's also very important that I make us go through some of, so you see me going down, I'm trying to check my document here. So also very important that I also let us know about how today's program is going to go, today's webinar is going to go. So we're going to be having 3 speakers on board. Why I'm going to be the moderator. We're going to be having, we have Mr. Geoffrey Otim. Mr. Geoffrey Otim, that's good to have you here. Welcome to today's webinar. We also have Dr. Nathan Hillson, and then we have Mr. Herbert Oloka. They're all going to be our speakers for today's webinar. So it's I'm going to introduce each of them when it is their turn, to speak. I'm going to read their bell. So that we don't spend too much time on introduction, we're just going to go straight to the webinar. So the first speaker for today's webinar is Mr. Geoffrey Otim, and then I'll quickly read his bio for us to understand who Mr. Geoffrey is, and also for us to understand his area of specialist. All right, so I'm going to start reading out Mr. Geoffrey Otim. I'm also going to spotlight him so that everybody can. Mr. Geoffrey, please, can you kindly turn on your video? Yes, please do turn on your video so that, yes, so that I can add it to my spotlights. Good to have you on board, Mr. Geoffrey. Thank you so much for joining us from Uganda. Can you hear me?
Yes, I can hear you. Can you hear me?
Yes, I can hear you. Amazing. Good to have you. So I'm going to be reading your bio for our audience to get to know more about you, and then before you go right into your presentation. So Mr. Geoffrey Otim is the founder and as the CEO of Symbio Africa. Symbio Africa is a forum for academics, for students, for scientists, for policymakers, and the general public to convey and develop effective pathway for development of synthetic biology across Africa. He has over a decade of experience working in the areas of molecular biology, medical laboratory technology, health management, biosafety, biosecurity, and international development with the International Food Policy Research Institute, Mary's Top International Uganda Virus Research Institute / WHO, and the United Nations Mission in South Sudan. Mr. Geoffrey has, over the years, Mr. Geoffrey has built a robust community of stakeholders in synthetic biology across the African continent, including founding the first HiGEM team from East Africa, which have organized the very first International Synthetic Biology and Biosecurity Conference in Africa. I'm also glad to let you know that Mr. Geoffrey has received several international awards. He has received several fellowships and invitations to speak on the topic related to synthetic biology and biosecurity at many international and local conferences in Africa, Europe, North America, and Asia. What a very rich bio. Mr. Geoffrey, it's so good to have you on GET platform. And then I'm also know with your bio, I'm sure participants are looking forward to learn so much from you. So Mr. Geoffrey is going to be talking on understanding the concepts, and Mr. Geoffrey is going to be talking on topic understanding the concepts and benefits of synthetic biology to promote sustainable development in Africa. So what I'm just going to do is that, Mr. Geoffrey, can you share your slides? Do you have a slide? Yes, I knew you can share a slide, okay? All right, so thank you so much for coming, Mr. Geoffrey. So I'm going to leave the scene for you to talk, to make your presentation.
So much for that wonderful introduction. Yes, I'm privileged to be talking today in this GET webinar series, March. Yeah, I'm still trying to set up my presentation. show. Okay. So I hope you can see my slide.
Yes, very well. I can see your slide.
Yeah, so let me first put it. Okay. Yes, so I've already been introduced, and I'm going to talk more about understanding the concepts and benefits of synthetic biology, and more especially in promoting sustainable development in Africa. Yeah, so as they have said, I'm the founder and CEO of SynBioAfrica, which is a synthetic biology platform for promoting the development of synthetic biology across Africa. So my talk, I'm going to, this is the outline of my talk. I'm going to go through an introduction and a brief overview of synthetic biology, not a lecture, but an overview, giving you some principles and enabling factors and applications of synthetic biology. I will also give you information about sustainable development in Africa, understanding how synthetic biology can be used to get sustainable development in Africa. I will also give you highlights of some of the benefits that synthetic biology has in terms of sustainable development in Africa. And I will also touch about some of the challenges that SynBio and sustainable development in Africa face. And lastly, I'll give way forward and recommendation. Yes, so briefly, this is what SynBioAfrica is a forum for scientists, researchers, policymakers, students, and the public at large to come together to harness this new synthetic biology science for the betterment and for the growth of, biotechnology advancement in the region, and also to tackle some of the challenges that we have in Africa. So our vision is to have a healthy, safe, and sustainable world through synthetic biology. And our overall mission is to be the leading SynBio innovators for sustainable solutions to great challenges that we have in health, agriculture, and environment in Africa. So this is the themes and the collaborators at our Center of Excellence that we are establishing. We are in the process of establishing. So the key themes that we have research and development, capacity development, where we run trainings of our scientists across Africa. We also have bioinformatics and data science. This is one of the key areas in synthetic biology. And we also have biosecurity and biosafety. You cannot do synthetic biology without having the aspect of biosafety and biosecurity. So it's very key. And we have Innovation Hub where we incubate some of the great ideas from our scientists. And the pandemic, the current or the recent pandemic has shown us that to address any pandemic, we need One Health approach. So that's why we have included One Health under our SynBioAfrica. So we collaborate with the public, civil society, educational institutions, private sectors, and the government, because we cannot work in silos. We need all the partners on board. So briefly, I'm not going so much into this because it's not our main focus. These are some of the milestones that we have so far. Yeah, we have so far run a global catastrophic biological risk initiative that we had workshops in all the 5 regions in Africa. And also we have run the first international synthetic biology and biosecurity conference in Africa way back in 2021. And that was the launch of SynBioAfrica. Others we can see we have emerging leaders that we sponsored way back in 2021, about 16 of them from 14 African countries. We have trained bioinformatics students or trainees, about 42 of them, and we have supported about other IGM teams from 4 African countries. So going back to the topics of today, I want us to start by understanding some of these key concepts. And yeah, so the key concepts that we are going to talk about is synthetic biology and sustainable development. So when we talk about synthetic biology, we are going to be discussing some of the cross-cutting applications of synthetic biology, especially in medicine. A lot of development has been coming through, and new medicines has been coming because of using synthetic biology tools to identify some of those novel biosynthetic genes. And also in relation to environmental protection, we talk about bioremediation. And in agriculture, a lot of technologies have been developed geared towards improving the quality or the yields of our agricultural crops. And information storage. Synthetic biology has also application in this area. Companies, big companies like Twist Bioscience have already piloted this, storing information in DNA. It's not a new concept now, it's being explored. And when we talk about sustainable development, we have three dimensions. We need to talk about it in the environmental, economical, and social aspect. So all these are enabling factors that we need to have in place for us to have a sustainable development. Yes, so still back to now defining what synthetic biology is. Yes, so when we talk about synthetic biology, first of all, the term synthetic, I mean, from de novo, synthetic from scratch. So that is how the definition came in. So in this case, we define it as the design and engineering of new artificial molecules, pathways, cells, tissues, or organisms. Or we can also redesign some of these biological systems for other useful applications. So as we can see from here, the molecules can be DNA, RNA, or proteins, and then you can be able to insert it in a vector. Like here we have can have a plasmid with sets of genes that can form a pathway that you have designed, a new pathway that you have designed. And of course, we have the cells that are already modified based on the design that you created from the first. And the cells can be modified to produce biofuels, and also can be modified to produce some of the pharmaceutical products. But the process takes quite some time using well-defined protocols. Yes, so we can also enhance tissues and the overall the cell organism. We can also enhance through these synthetic biology technologies. And we have seen humans are also part of this engineering process, but at the moment we still don't have the law that permits us to do the engineering of a human genome. Yes, so this is giving us all the overviews of why we are talking about synthetic biology and why it is very important for us to harness this new science. So the key principles that it works on is based on, as I told you before, you start by design, the design, build, test, and learn. You design what modification do you want to do? What do you want this biological system to do for you? You want it to produce for you biofuels. You want it to produce for you new bioactive metabolites. So it's upon you to design, and this is usually in collaboration with computational biologists. They are more well-versed with this design phase. And after that, having all your design in place, you can now do the building, the construction of your biological system through the vector system, and also can be through other methods. So after having all your construct in place, you have to test to show that, to prove that it is really producing what you had intended it to produce. Like if you wanted to produce a biofuel, is it producing the biofuel of the quality and the standard that has been designed for? So after that, you can also learn from the results that you have got. And if it doesn't work, you go back and again modify some of your pathways so that you are able to produce the expected product. So in doing that, we already have some of the enabling technologies or some of the factors that makes synthetic biology, feasible. So overall, you already know that we have a very inexpensive DNA synthesis at the moment. Before it used to be very expensive, but at the moment there are so many companies that are doing gene synthesis. So the price has really come down very fast, and that is one of the factors that enabling these technologies to go on. Yeah, so there's a lot of improvement in DNA and genome sequencing. Yeah, so many robust methods have already come up to do this sequencing, and it takes literally a shorter time like before that you used to send abroad and you wait for like a month without getting the results. So it is also one of the enabling factors for us at the moment to enhance synthetic biology. Yeah, we have already DNA assembly methods. We have Gibson. We can also use the PCR-based approach to do this. The assembly and so many other methods that are currently in use and very robust for doing assembly. And we have pathway engineering. You can engineer your pathway in a vector, and then you can insert it in a biological system that you want to engineer. Then genome editing, we already have CRISPR technology that is having a potential even to be applied in sickle cell disease, gene therapy. And also genome shuffling, this one you're trying to combine genome from different strains so that you come up with a more improved phenotype. Yeah, so basically that is what we have at the moment that enables us to harness this synthetic biology and to apply it in different fields. We have health. We can apply it to get new antibiotics, also to come up with new vaccines, RNA vaccines that are currently being used for COVID came because of some of the technologies within synthetic biology. And also having safe food, because we can also apply to enable us get quality and enhanced yield of foods that we have. Yes, so in the environmental aspect also, it can be used in bioremediation, and in areas of energy, we can use it to produce biofuels using the available biomass that agricultural biomass that are everywhere. Like in the African context, we have biomass everywhere. And yes, so when we talk about sustainable development, we are talking about a development that meets the needs of the present, the needs of today. But it should not compromise also the ability of the future generation to meet their own needs. So this is key when we talk about sustainable development. And this is anchored on the 17 sustainable development goals that the UN came up with. And these 17 sustainable development goals, we can achieve it through these 5 principles. First of all, we have to have, we need to live, this is the principle of sustainable development. We need to be living within environmental limits. We should not encroach on some of the protected areas because they are there for the purpose. Like if we deforestate, If we cut down forests, definitely we are going to get a problem with a climatic problem. So we should not encroach beyond our limits. And also we should live by achieving a sustainable economy where everyone can survive, economy where everyone has equal right to access some of the services that are needed for their livelihood. And also promoting good governance. You know, our economy relies mostly on the political will or the policymakers. They need to have good policies in place so that we are able to gain all the livelihood support that we need. Yeah, so also using sound science responsibility. Our scientists should also use science responsibly. I talked about biosafety, biosecurity issues. You don't need to do science while it has environmental impact, or it can also encroach on human life. So you need to have all these aspects in place. So also ensuring strong health and a just society. This is one of the principles for us to have in place in order to go forward to sustainable development goals. Yes, so this is now the summary of the contributions of synthetic biology to the sustainable development goals that the UN has come up with. So here it touches on some of the sustainable goals, not all. The first one is clean water and sanitation. So these are some of the innovations that have already been done or are also being done at the moment. Like the innovation of having a water filter made of wheat proteins and potato starch that incorporates bacterial monooxygenase to remove benzene from contaminated wells. This usually is applied when there is any oil spill. So in terms of clean water, this is some of the innovation already being done. We have decent work and economic growth, which is sustainable development goals number 8. So this has already been, a startup in Kenya uses synthetic biology also to produce a high value compound from the over harvested plants. So this is a startup in Kenya already using synthetic biology. We have industrial innovation. This one is trying to replace the dye using so the dyes that are produced from silica, which is now a new dye that are being used, is being used to replace azo dyes that are used in textiles. So it's one of the innovations within the synthetic biology space. And we have reduced inequalities. So here, when we talk about reduced inequalities at the social aspect, we talk about at the moment we have the Nagoya Protocol on Access and Benefits Sharing. We already know there are a lot of genetic resources in the environment there, and some of these genetic resources are under the control of the indigenous people. So there should always be a benefit sent back to the community, the indigenous people. So this is why we are talking about reduced inequalities by giving back royalties to such indigenous people. Sustainable cities and communities. We have also responsible production and consumption. Yes, so here we have industrial chases that uses synthetic pathways for waste valorization. Sorry. Yes, so this is trying to get another source of energy instead of using the glucose and nitrogen is also another innovation we have. And life below water, trying to protect the humans because we depend on fish that are found in the underwater. So there are always some contaminations that usually we consume from these fish. So there are innovations that are using biosensor that can detect some of those contamination, and appropriate approach can be used to address. Yes, so also we can use synthetic biology to address some of the issues around life on land. And so bacteria and plants that are engineered to remove polyaromatic hydrocarbon and polychlorinated biphenyls from polluted land. This is bioremediation that I talked about. So trying to have a clean environment free from pollution. Yes, so when we talk about some of the challenges that we have for us to move forward to a sustainable development in Africa, we have the challenge of implementation of some of these programs, and this implementation should be aligned with each country needs, because what can be implemented in another region may not be fit in another region. So it should be well aligned with the local context. And we have governance, but most especially the political will. Political will is very key for us to go towards sustainable development, because some of the enabling policies that we need for the technologies that I've talked about to flourish may not be there. So you cannot be able to use such technologies. And poverty is well known that it is one of the major issues that constrain our social and health impact. Yeah, so if you don't have the basic needs, or you don't have money to access health services, of course, this is going to be generalized and contribute to low sustainable development in a particular country. And also we have climate-related disasters. We have drought and desertification and social underdevelopment. So these are some of the key elements that contribute to underdevelopment. Yes, so in real life, we have seen that some of the technologies that I've talked about have very negative perception in the African perspective. We already have the expertise that are well trained to do or to carry on with some of these technologies, but the perception outside there is really very negative, and the support for these kind of modern technologies like the CRISPR-Cas technology that I talked about, it has a lot of negative perception. So we need to move forward and see how we can support this. So also synthetic biology in the African context has a lot of challenges. First of all, we don't have the policy to regulate synthetic biology and biotechnology in Africa, which is one of the key bottlenecks that we cannot move forward. And we have a very negative public perception towards biotechnology. When you talk about biotechnology, the only understanding is GMO, which is not the case. There are a lot of other applications of this biotechnology. I've talked about health and also environmental bioremediation. So we currently we don't have national strategies and roadmap for synthetic biology and biotechnology in most of the African countries. So this translates into lack of funding, because if we have a national strategy for synthetic biology, definitely it has also to have a share on the national government budget, but that has not been put in place, so we still have that challenge. And we don't have, at the moment in the African institutions, we don't have any courses, whether undergrad or postgrad programs in SynBio. So we need to work towards having this put in place. Yeah, so this is my recommendation based on the challenges and the promises that synthetic biology has towards sustainable development in Africa. So we need to have a regulatory framework to untie the hands of the scientists to exploit their potentials, because they already have the expertise needed to run some of this biotechnology work. So we need regulatory framework in place. And also we need to develop and effectively implement the national roadmap for synthetic biology. Yeah, we also need to, advise some of these policymakers of ours to go for benchmarking visits and listen from other policymakers how they have managed to have a policy to regulate some of these biotechnology related advancement. And inclusion of synthetic biology in the national annual budget that I've talked about, so that we have the funding to support some of these innovations. And the involvement of private organization and civil society organization in government programs. In most cases, civil society organization are left out. Government prefers to work in silos, which is not a case. So in this case of policy and decision making, everybody should be on board to make a very informed decision. And international collaboration and funding support also is one of the aspects that we need to work on to support the budget and the technologies that are needed for us to move forward with synthetic biology. Yes, so all this information was got from the references I've put here. We also have a publication on the SynBioAfrica story from the grassroots, the present, and the future. You can access it from biotechnology notes and other references. Yes, so this is now some of the upcoming projects that we have at SynBioAfrica, and we want it to be spread across African countries. We are going to start from Uganda and see which other country can accept this, and then we shall come and see how to implement. This is more of bioprospecting. We are going to go conventionally, because we know that there are a lot of silent biosynthetic gene clusters that are still in the environment, and this we are targeting streptomyces because most of the antibiotics that are on market were got from streptomyces. So we are going to do the soil sampling. We isolate the streptomyces. We do the DNA extraction, and then we do the PCR to amplify the DNA that we have extracted from the soil sample. And then from there, we do the general sequencing, because we want to know what sequences do we have still in the environment there. And after doing the sequencing, we look for those unique biosynthetic gene clusters that are silent, not usually expressed in the laboratory using the normal laboratory procedures. Yeah, so these are some of the hidden biosynthetic clusters that could be a potential drug lead that we are trying to look for in the environment there. And from there, we can go forward into analyzing it and assessing its suitability to be an antibiotic. So that is one of the projects that are coming up. And yes, we also have the second International Synthetic Biology and Biosecurity Conference in Africa that is happening this year from 12th to 14th July 2023. So all of you are welcome. Look at our social media platforms for information about this conference, and then you can be able to register for it. The key themes will be innovation in agriculture, bioinformatics and AI, biosafety, biosecurity, renewable energy and materials, then biomaterial science and biosensor, including diagnostics. We also included One Health because we are still in this in the period of the pandemic. So we want to bring on board different partners and people who have responded to pandemic to share their experience. Yes, so that is basically what I had for you. And I thank you. These are the partners we have worked with so far in the 5 years that we have been in existence, SIN by Africa. So we are also happy to collaborate with GET consortium to organize this webinar. Thank you so much. And back to you, the moderator.
All right, thank you so much, Mr. Geoffrey. Thank you so much for that very enlightening, very educative presentation. I'm just going to run through some of the things that you mentioned, just to do like a recap. Once again, I wanna commend you for that amazing presentation. So from your topic, you've been able to highlight, educate us about the amazing benefit that lies within synthetic biology. Okay, so I'll just so like we can both see our face. Yes. All right, so some of the things I had there, I'm looking at my notes now. You mentioned that synthetic biology can promote technology in several ways. And I recall that you mentioned that through synthetic biology, some of the medical practices, some of the biological laboratory practices have been made inexpensive in Africa. You did also mention that synthetic biology has promoted a lot of innovation in the health sector, which include production of antibiotics, vaccines, and gene therapy. You did also highlight the important role or important benefits of synthetic biology in the food sector, which also include promotion of food safety. You did mention about the role or the benefit of synthetic biology with respect to agriculture, to energy and more. And then I also love the fact that how you were able to bring together, or how you're able to sync synthetic biology with the Sustainable Development Goals. And I saw that you highlighted something here that through synthetic biology, we can live, the environment can be safer, we can achieve a sustainable economy. I think I did mention during my introduction that synthetic biology has been globally recognized as one of the fundamentals for bioeconomy. And then your presentation your presentation also emphasized that. You also did mention that through synthetic biology, bringing synthetic biology and sustainable development together, science can become more responsible. You did also mention, you highlighted some contribution of synthetic biology to UN 2030 sustainable growth, of which you highlighted some key points, which include through synthetic biology, we can achieve clean water and sanitation, we can achieve decent work and economic growth, we can achieve industrial innovation, we can also reduce inequality such that both the international and the local innovation can be brought together to work together. And then I also love the fact that towards the end of your presentation, you did make way forward and recommendation, and of which we're also going to package and share with our policymakers. You mentioned that it is very important that we begin to involve private organizations, CSOs, and also CSOs in government policies and programs. You also mentioned that it's very important that policymakers come to recognize the concept of synthetic biology. And I'm also glad to let everybody know that the March edition of GET webinar series is being organized in collaboration with SynBio Africa. And then it's so amazing to also know that you, towards the tail end of your presentation, you made everybody anticipate the second International Synthetic Biology and Biosecurity Conference in Africa that's going to be organized by SynBio Africa in Uganda. Thank you so much once again, Mr. Geoffrey, for that enlightening and educative presentation. I would also like to tell our audience that please kindly use the chat box in case you want to drop your name or where you are actually joining the webinar from. And it's also very important to let us know that some of us are joining, and you can see that when you join, your username is showing webinar participants. It must have been because you did not join with the personalized link that was shared with you, but no worries, we got you covered. Kindly click on your name, on the username on your phone or on the devices that you're using. You can easily edit it and change it to your preferable name. And then we also have a question and answer box right at the bottom of your screen. You can drop your question. I can see that we'll be receiving questions, and then by the end of the presentation, when all of our speakers have made their presentation, we'll go back to the question and answer section for them to do justice to all of the questions that we've been bringing. All right, so I'm just going to go straight. Thank you so much, Mr. Geoffrey, once again. What I'm just going to go is that I'm going to go straight to the next speaker. So I'm going to be spotlighting Dr. Nathan. So please, Mr. Geoffrey, I want you to be on standby because you're going to be answering some questions from our audience very soon. Thank you so much. All right, so I'm going to be spotlighting Mr. Geoffrey, Mr. Nathan, rather, Dr. Nathan, please pardon that. Dr. Nathan, it's so good to have you here. I hope you can hear me.
I can hear you. Thank you.
All right, amazing. So before I actually give you the platform, I would like to read your biography so that our audience can get to know you more and get to know what you're doing. Dr. Nathan Hillson earned his PhD in biophysics from Harvard Medical School. He did his postdoctoral work in developmental microbiology at Stanford University School of Medicine. Dr. Hillson work has spanned the realms of the private, notably as the co-founder and the chief scientific officer at Tesla Gen Biotechnology Incorporated and public biotechnology sector. Dr. Nathan, he as the department head of biodesign within the biological system and engineering division. Dr. Hillson leads scientists and engineers within Lawrence Berkeley National Laboratory, whose domain expertise spans synthetic biology, metabolic engineering, microbiology, microbial communities, software engineering, and laboratory automation engineering. As overall principal investigator of the US DOE Agile Biofoundry, Dr. Hillson leads an even broader group of scientists and engineers distributed across 8 US DOE national labs towards development of the public infrastructure that enables the private sector to reduce the cost and accelerates bioprocess commercialization timelines. It's also very important to let us know that Dr. Nathan is the director of synthetic biology informatics at the Joint Bioenergy Institute. It's so good to have Dr. Nathan here on GET webinar, and we want to appreciate you for accepting our invitation. I will also let everybody know that I would like to also tell us the topic Dr. Nathan is going to present it on. Dr. Nathan is going to be talking about identifying the potential risk and consequences of synthetic biology on human, on animal, and environment. So it's so good to have Dr. Nathan. I'm just going to open the floor for you to carry on with your presentation.
Well, thank you very much for that introduction. I would like to thank you and the other organizers for the invitation to be participating in this GET webinar. It was also very nice to be able to follow Geoffrey. Geoffrey gave a very nice presentation that basically not only talked about some of the details of synthetic biology and its benefits, but also making sure that we do it in a thoughtful and sustainable way. So I think it's nice that my kind of presentation will follow Geoffrey's talk. I do not have any formal slides that I'll be presenting, so I'm just going to be speaking to you. I will be sharing my screen here in a second to show you some things that I think are very relevant to risk assessment in the engineering biology or synthetic biology space. And after I give my remarks, I'll be happy to look at the Q&A and answer any questions there, or if there's any that fall out in the webinar. I'd also just like to point out that in California, it's currently 4:50 a.m. So I'm definitely in the middle of the night here. So hopefully I'll be coherent and be able to say some things that are helpful and useful to this community. So I guess one other thing that I'll just mention, and then I'll come back to it at the end, is that in Geoffrey's presentation, he talked about the public and how they view synthetic biology and other types of things. And I will come back to that at the end. I think that's an important thing to talk about. So we'll get back to that in a second. The other kind of just little preamble that I'll mention, oftentimes when people are talking about risks, they largely fixate on negative risks. So if you do something, something bad might happen. But I would also just like to remind people that if you do not act, your inaction also has a risk, and that you are giving up a potential benefit of having done that action. So risks aren't only about, if you do something bad will happen. It's also, if you do not do something, then you might be missing out on the opportunity to experience that benefit. So risks can be both positive and negative. The other thing here is that the conversation around risk and engineering biology is a very, very, very large one. And I will not be able to do it justice in the few minutes that I have with you. I would also mention that I have been trained on the scientific side. I have not been trained on like the risk assessment side of things. So just to point that out, I'm going to go ahead and share my screen. And I want to point out to everybody here on the webinar a resource that I think is going to be very nice and helpful and timely to folks. It might be difficult for you to see the URL. So I'm just going to bring up a text editor. So this, and I can also put it into chat. Maybe I'll do that too, so people can just have the link, but I'll just share it on the screen here too. Let me paste it into chat. So that's a link for everybody. But I'm going to be showing you a little bit from the Stanford Biopolis site. And in particular, there was an effort led by Stanford that was supported by the Nuclear Threat Initiative. That's kind of has the acronym VEERS, but it's a visibility initiative for responsible science. And if you all go to this website, one thing that you can do is look at basically a PDF output from this project, and it's a 91-page document, and it's all about biorisk management, and it's effectively a case book. So it has a whole bunch of kind of like case studies around the practices for assessing and managing the risk associated, for example, with synthetic biology. So I think this is a really great free public resource for people to look at. And one thing that I would mention is that a lot of the experience that I'll be telling you about, not all, but a lot of the experience that I'll be telling you about is in one particular case study that comes from the U.S. Department of Energy's Joint Genome Institute. And this is the case study that I and other people associated with the Joint Genome Institute contributed to. So if you want to see written documentation around what I'm going to be sharing with you, this will be a good resource for everybody to look into. So I'm going to go ahead and stop sharing my screen, because the rest of my time will just be with kind of just oral remarks. When you're talking about synthetic biology, as with any technology, it's very important that the people that are planning future work, research and development, other types of activities, are not only concerning themselves about all of the scientific knowledge or the technology that could come out of these efforts. It's also very important to consider the broader aspects and implications of the work. So if you were to do this research and development activity, or you were to launch this technology out into the world, what would that world look like? What would be the consequences? Those could be to humans, it could be to animals, it could be to the environment. There might be social justice types of aspects to this. So social justice meaning that anytime something happens, there could be certain people that would benefit, but there might also people that would not be as well off. So say, for example, you're talking about, I think in the end of Geoffrey's presentation, there was some really interesting discussion around trying to mine the soil for natural products, for example, and these could be used as medicines. If you're going to be using synthetic biology to produce medicines in a kind of a new way, that might be affecting people that are producing medicines in a more traditional way. So even though maybe large portions of the population or the world might benefit by certain innovations, there might be certain groups, for example, maybe farmers that are growing crops and extracting medicines from those crops, that might be very intensely disadvantaged by those improvements. So it's very important when considering kind of research and development things to more broadly consider the world that you would be enabling or creating should your science and technology development be successful. There's also legal types of implications of these works. There could be governance and policy types of implications. There's lots of different aspects to the research that we do, and lots of scientists are not necessarily experts in all of these different domains. So it's very important to make sure that even when you're at the early stages of science or technology development design, that you're thinking broadly about what these types of implications and aspects might be, and that you have people to talk with that might be much better experts in these different topical areas than you might be. So I can tell you, some of the things that we've been doing, for example, at the Joint Genome Institute in the US, as part of its synthetic biology type of user program. So the Joint Genome Institute is a user facility where anyone in the world, including in Africa, and everyone on the call is included, can submit proposals to the Joint Genome Institute, basically for the Joint Genome Institute to help enable your science. And that's available at no cost. It goes through a competitive selection process, and there are evaluations on, is this work going to be relevant to the Department of Energy and those types of concerns. But almost, all of the proposal selection process of the Joint Genome Institute goes through this competitive selection process. And usually it's for assessing things like scientific merit, but also Department of Energy and energy relevance. A number of years ago, when the synthetic biology program was just launching, and Eddie Rubin was the director of the JGI at the time, Eddie Rubin was very excited about announcing the synthetic biology program publicly and wanted to let people know about it. And this was done at an annual meeting that the Department of Energy puts on for people that are supported by its biological and environmental research sub-office within the Office of Science. And at this meeting, Eddie was presenting about a potential technology for partially rewriting the genome of a bacterium that would be useful for a biomanufacturing process. And this might nowadays be using technologies like Geoffrey mentioned with CRISPR and other types of approaches. But the basic concept was that if you have an industrial process for biomanufacturing using a bacterium or other types of organisms for producing things, for example, through fermentations or other types of bioreactor types of production processes. A challenge with using bacteria is that they're susceptible to phage infection. And I think I saw on one of Geoffrey's slides that you can use phage as part of like a cosmetic type of a therapy. So maybe there might be bacteria causing acne, and you might think about using phage to kill the bacteria that are causing the acne. And anyways, in an industrial process for fermentation, if you get a phage contamination or a phage infection in your bioreactor, it can spoil that entire batch. And that can cost you a lot of money and a lot of time, and it's can be very time consuming to make sure you clean your bioreactor to make sure it's sterile and doesn't have any traces of the phage infection. So it's a big problem industrially. So one of the projects that Eddie was talking about in this announcement of the JGI synthetic biology program was, one project that you can imagine is partially rewriting the genome of the bacterium so that it is no longer sensitive to phage, so that you don't have to worry about your industrial process getting shut down by a phage infection. And this sounds great and very relevant. But after the presentation, Eddie received a question from someone in the audience, and the person in the audience asked Eddie, this all sounds great, but what happens if your engineered organism gets out into the environment and it is no longer phage sensitive, so it doesn't have its natural predators that would basically make sure that it doesn't spread? And Eddie hadn't really considered that type of a question. He wasn't really prepared for it, and didn't really give probably the best answer to that question. Now Eddie had a lot of experience in animal research and had lots of experience with institutional review boards. And coming out of that experience, Eddie came back to the JGI and told us, we really need to have something like an institutional review board that's done for human or animal studies, but we should be doing something similar to that for synthetic biology or for engineering biology. So out of that, we developed this process for beyond just the scientific merits and DOE kind of relevance of projects at the JGI, at least for the synthetic biology program, to also have a review process that's including domain experts across biosecurity and environmental issues and social justice and governance and policy and legal aspects, ethical aspects, to evaluate these proposals to help us think really through the consequences of these types of projects should the JGI select them and actually enable them. And that's been fantastic, and a lot of kind of insights that I can share with you come out of that experience, and that's what's kind of documented in the documents that I referred you to and kind of pasted in the link for you to read more about. And maybe just some examples besides the phage resistance type of a topic. One example of a project that I think is relevant is that there was a group of scientists that were very interested in understanding how a rice pathogen was infecting the rice plant. And they knew that the rice plant was responding to the presence of the pathogen and had its own immune response. They wanted to understand that a bit better. And they had some questions, was the rice plant detecting the pathogen just based on physical contact? Was it based on some type of a chemical signal that the organism was giving off? How did this all work? With the idea being eventually you could potentially work on the rice plant to make it more resistant to these pathogens. But what they had proposed to do was to actually engineer the pathogen to be able to have it not emit this chemical signal that maybe the plant was detecting. And that way they could assess, like, how does the plant respond with this chemical signal present, and how would it respond without that chemical signal being present. But what they didn't totally think through was the fact that they're effectively making a stealthy rice pathogen. Because they're engineering a rice pathogen that no longer emits this chemical signal, so effectively it's very stealthy. So now, of course, rice feeds a lot of people around the planet, and this rice pathogen was already a select agent and something of concern, and they were going to potentially make it more pathogenic. So this was definitely something of concern, and this was something kind of identified out of this broader aspects review process. As it turns out, it also would have been flagged. Geoffrey, in his presentation, he mentioned there's lots of DNA synthesis vendors out there now, including Twist, but other companies. And many of these companies have joined the International Gene Synthesis Consortium, and they actually follow very good practices around screening their customers and also screening the DNA sequences that they're providing or generating for their customers. And we also use those types of practices at the Joint Genome Institute. So even without this review process, we would have known that this particular group was interested in engineering the rice pathogen. But I think this broader aspects review process was very helpful because we started to understand the intent of what the researchers were doing. So we knew that it was valid research process, and through back and forth with those researchers, we understood that they were actually working in a biosafety level 3 laboratory environment. They were already properly licensed to work with the select agent. Everything was above board, everything made sense, and everything was going to be done properly. But what was interesting was that the researchers themselves hadn't thought more broadly about the potential impacts of what they were doing. That's one particular example. Other types of examples to kind of mention, for example, might be there have been projects where people were sourcing genes from all across the tree of life. And occasionally, some of those genes come from select agents, so organisms of concern. And oftentimes the users aren't necessarily thinking so much about the source of a gene. They just kind of are looking after the function. And oftentimes they're perfectly happy choosing alternative genes that don't come from organisms of concern. So that's something that can be done that's helpful. And it also sometimes will happen to lead us to some more types of ethical types of questions. So occasionally, some of the genes that people are going to be using for engineering purposes might actually come from humans. And are you introducing human genes, into microorganisms, including yeast, for example? And there might be some ethical, types of concerns around that. I think there's other types of stories that I can tell. Some of them will be in the document that I referred you to earlier. One other type of thing to mention, I think, is as people are working on tools and capabilities for synthetic biology and making things available that can enable other people. In my particular group, we, amongst other things, we do develop software platforms that can enable people to do engineering biology, synthetic biology more effectively. And it's very important, based on our experience that I'll tell you about, to make sure that you screen your users and have a somewhat of a sense of what they're going to be using your tools for. So just one anecdote, a couple of years ago now, there was a person that had requested an account on one of our software platforms. And we, as part of our process, we want to make sure that each user is affiliated with an actual institution, whether it's a university or a national lab or a company, some entity that would have some type of oversight, whether it's biosafety or ethics or proper laboratory types of activities. And this particular individual that was signing up for an account was just an individual, had no training in biology or science, was not associated with any institution that was relevant. And trying to vet this account, I had some email exchanges with this person, and it turned out that this person was in a crisis situation for his family, and his wife had developed a very rare genetic disease, and there weren't any available treatments or therapies. And his intention was to engineer a bacterium to actually do a genetic therapy treatment on his wife. So he was going to be using our tools to engineer an organism to do gene therapy on his wife, when he had no training, no institutional oversight. And this wasn't really something that we wanted to enable. So we did not enable his account. And we also referred him to others with domain expertise in this to kind of talk through what he was thinking about. So that's just another example. And then maybe just one last thing before I conclude. And I probably have gone over time, and please chime in if you want me to wind down even sooner. But one other thing that Geoffrey mentioned in his presentation was around public perception. And one thing that we've done, I think has been very helpful, is going out into the community and having open discussions about what we're actually planning on doing, and how we think about those things. And I think when we've gone out into the community and we mentioned that, not only we're doing the science, but we have a process to, at the very early stages of experiment design, that we're thinking broadly about the impacts of what our work might come to. And this is kind of how we're doing these things. And we're not just giving lip service to the fact that we think that these risks are important to consider, but we're actually doing something about it and investing our time and resources into making sure that we're going to be making for a better world. Then I think that's largely helpful, not only in terms of public understanding, but having them understand that you're actually trying to be doing these things in a sustainable and responsible way. So I'll end there. I apologize if I've gone over time, but I hope what I've said has been a little bit helpful. So thank you very much.
Thank you so much, Dr. Nathan, for that rich conversation that you has pulled us into. Yeah, your presentation was very timely. So, and then I want to also thank you for taking out the time. I know that you earlier mentioned that the time is going to fall into the early morning, and I want to thank you for taking out of time. I'm sure we will actually shorten your break time. But thank you so much for that sacrifice. Thank you so much. I can see a lot of questions that have been dropping in our box. I'll just do a little recap, just like I did for Mr. Geoffrey, of what Dr. Nathan's presentation was centered about. I'm really excited that there is a lot of connection between the first presentation and the second presentation. And I really enjoy the fact how you were tying it together, and that actually made the webinar quite a very smooth conversation. And I also love how you started, that you said that do not do anything is even risky. So it's so important that you want to do something. Then I also love the fact that you were able to give practical examples of what is going on in the line with scientific manipulation, demonstration, and innovation. And then I love the fact that you were literally pointing out that there's a need for regulation in all of this activity, especially because there are some scientists or there are some more like the innovators that are actually not scientists in particular. Thank you so much, Dr. Nathan. I'll just like I said to Dr. Geoffrey, I would like you to be on standby. We're gradually wrapping up the webinar, Well, thank you so much. So I will just go straight to continue to bring up our third speaker. But before I do so, it's also very important that we take our time to appreciate our audience. I can see in our chat box, quite a lot of people have been dropping their names and where they are joining the webinar from. I'll just quickly take about 1 to 2 minutes to just take a note of all this, because one thing about Get webinar, what has made us stand the time, we've been organizing this webinar for over a year, and the beautiful thing is that we've been having a diverse community of audience coming to join us. Currently now on the chat box, I'm looking at my phone and I can see Samira. Samira is joining from Morocco. We have Faith joining from Nigeria. We have Abeniza, sorry if I don't pronounce your name very well, please do pardon me. Abeniza is joining from Ethiopia. We have somebody joining from Morocco as well. We have another person joining us from, someone said, hello everyone, my name is Salahu Idris from the Ministry of Foreign Affairs Research Department. It's so good to have you. We have Rurala, joining from Ibadan, Nigeria. We did also have Antonio Callisto joining from UK, JAPSCO Limited. We have quite a lot of people joining us. We have somebody also joining us from Jude, joining us from Uganda. We have somebody joining us from Pakistan, Hamba from Pakistan. We have another person joining us from, we have a PhD student, big girl. Sorry, I do apologize if I don't pronounce the name well. Is joining us from Ivory Coast. We have somebody joining us from Charles. We have Charles joining us from Kenya. And we did have Otim joining us from China. We also have another person just joined and said, okay, my name is Alo, I'm joining from New York. You can see that I've actually confirmed what I've earlier said, that every month we have this webinar, we always do have a diverse audience. So I'm also aware that a lot of people are dropping comments in the chat box. Please, our time may not permit us to go over your comment, but I'll do look at how I can read out your comments. But most importantly, please do drop your questions in the Q&A box at the bottom of your screen. So I'm going to go straight to our third speaker. We have our third speaker here. Our third speaker is Mr. Herbert Oloka. Before I actually bring Mr. Herbert here on board, I would like to quickly read his bio so that we can also get to meet Mr. Herbert officially, just as the way we met all others or our other speakers. So Mr. Herbert, please, if you can hear me, kindly turn on your video so that I can also spotlight you alongside my video. All right, so I'll be waiting for you to turn on your video. So I'll add it to the spotlight. Okay, so I can see that. Oh yes, so good to have you. Welcome, Mr. Herbert. Hi. We're so excited to have you. All right, so let me just quickly go over Mr. Herbert's biography. Mr. Herbert Oloka, I do hope I pronounce your name very well, but please do correct me. Okay, thank you. He's a biosafety and bioscience development specialist, currently coordinating the efforts of programs for biosafety system in Uganda. He has specialized in agricultural and science policy, working with public and private sector organizations for 19 years. Mr. Herbert continues to work with national and international organizations in furthering food security, enhancing livelihood, and building resilience among communities in Uganda and in other African countries. He has actively engaged in biosafety policy discourse and technical capacity development for 15 years, supporting evidence-based policy and technical decision making on the use of modern biosciences, especially genetic engineering. His current passion is enabling innovations in modern biosciences to work for Africa. I think I love the fact that this is good. He is skilled in policy development, science regulations, plant breeding, genetics, and communicating science for impact. It's so good to have you, Mr. Herbert, joining us. And then by our schedule, Mr. Herbert will be making the presentation on promoting ethical and responsible use of practices of synthetic biology. All right, so Mr. Herbert, so good to have you. And I'm going to leave the floor right now for you to share your presentation and your thoughts with us. Thank you.
Thank you very much. I hope you can hear me well.
Perfectly, I can hear you so well.
All right, thank you. So I think mine will pick up from the previous two speakers, Geoffrey and Nathan, who highlighted, of course, the potential impacts of this technology, the benefits, and also Nathan, who talked about the potential risks in various areas. Fortunately, both of them talked about some areas of concerns and how these can be managed with some examples. And so I don't have a slide prepared for you, but I have some notes that I will be sharing from. And I do appreciate if you have any questions, of course, I'll be here as well to take and answer them. Yeah, so as the two have spoken, now when you come to how do we promote ethical and responsible use of synthetic biology, if you follow from Nathan's talk, it doesn't have to be about synthetic biology or biotechnology, it's technology in general. And we, of course, mindful that synthetic biology offers, a different set of tools in different fields that can result in products that combine different areas of or the biological disciplines or the physical sciences discipline, the chemical sciences discipline to create the same product or one product in that case. So all that brings in ethical dimensions or safety dimensions that people are not used to yet, and we have to start by maybe educating them. So we'll go through some of these notes I was saying, and I say that because it is just technology, any new technology will require careful and balanced consideration of the potential benefits and risks to humanity and the environment, and of course, the environment includes animals that are living with humans, or those that live outside humans. Of course, the environment will also include all microorganisms and everything else. So we've had lessons before from the pharmaceutical or health applications of modern biotechnology, as well as agricultural applications. For agricultural health, particularly maybe 45 plus years now, and then agriculture, if you talk about GM crops, 20 25 to 30, coming 30 years soon. And those applications or those developments have shown that careful and systematic and demonstrable consideration of the risks of these technologies versus their benefits during the regulatory process has allowed society, our current society, to benefit immensely from these innovations in the biosciences. We cannot overlook the power that the vaccines have produced today, or agricultural technologies that have enhanced yields across the board. So synthetic biology simply extends these considerations, and depending on the exact combination of techniques or combination of technologies or products, but also, of course, mindful of the deployment environment. Where will this product be deployed? Is it in a human body, or is it in an animal body? Is it in the environment of agriculture, environment of a forest, or is it going to be in a factory? But for what? It will still end up in a human body or an animal body, or part of the environment if you're doing bioremediation or any other environmental control approaches that Geoffrey mentioned earlier. So we'll definitely ethical and responsible use of technology. It's based on the principle of first do no harm. Yeah, before you start doing something, yeah, can you minimize harm to society? And that society, as I've said, will include humans or humanity, animals, and the greater environment. We have to be always mindful of all those dimensions. And of course, it's impossible to cause zero harm as you do anything in the world. It doesn't mean you can be very, very minimal risk to the world, but it's impossible to say there's zero harm, because in most cases, the natural deployment of any innovation may hurt a component of the environment to some extent, maybe a small extent or to a great extent. Yeah, but now what we should always remember, like what this emphasizing this from Nathan's talk, we should make sure that the benefits are greater than the harm. So our ethical discussion or ethical considerations must be that whatever whatever we intend to use technology, or whatever technology we have on the table, including synthetic biology, should be more beneficial to the society than harm society. And of course, as I will keep emphasizing that, because of the multidisciplinary nature of the synthetic biology applications and the multiplicity of the products, which combine biological, material sciences, physical sciences, other engineering disciplines, the considerations are really, really greatly broadened. And we must ensure that we integrate these considerations in our research and development activities involving synthetic biology. So how best can we promote ethical considerations in the use of synthetic biology? I feel there are just about three major approaches, of course, with many nuances in between there. But the first one is government-based regulation of the practices and processes and products. Prior to deregulation, prior to a product of stability coming to the market, there can be a set of government rules that somebody oversees your technology, your work, and make sure that the product is assessed for safety, assessed for ethical considerations before it gets to the environment. And we use ethics here broadly to mean cultural beliefs, religious, or any other, but as well as safety itself to humans, to livestock, to other animals, and then to the general environment. And we've seen this government regulation being very effective in other biosciences fields, especially pharmaceutical, agricultural, biotechnology, where GMOs have been released for many years without significant environmental harm or human health harm, not recorded. And then we've also, of course, seen many industrial bioproducts coming up, including enzymes we use in detergents, or living cultures of microbes that we use in like yogurt manufacture, or enzymes we use in food processing, like cheese manufacturing. And we've seen, that government regulation through standards and through laws has worked well. And of course, laws are always the first point of government regulation. So most people will work towards a law in place. And the world over Africa-wide, I think only Nigeria has so far established a clear law that they will be addressing issues of synthetic biology through regulation, through a clear system that will work with their National Biosafety Management Agency. Yeah, at international level, of course, the national laws are the best. Are always the basis, but sometimes they're backed up by international treaties that reinforce or establish an operating ground between countries, among countries. And at national level, we've seen the Convention on Biological Diversity take interest in the matter and that is through the UN system. And their most recent decision in 2022, December, was to allow the world to continue broad and what they call broad and regular horizon scanning, monitoring, and assessment. They want to continue understanding what is going on in the field, the new technologies that are developing, and they also established an ad hoc technical working group to make sure to do this scanning and reporting to other countries that are parties to the Convention on Biodiversity. And the aim here, of course, is to also promote information exchange among these countries on the development of synthetic biology, as countries think about how to regulate these technologies within their current biosafety context or establish new regulatory processes altogether. Yeah, of course, through the government laws of national laws, you can have subsidiary legislation like the regulations that are clear and tell you what to do during research, testing, and deployment of products. You can also have standard operating procedures that guide day-to-day operations of scientists and researchers. And the regulations, as I've said, might cover ethical considerations broadly or narrowly as safety considerations during this work, safety to the researcher, to the livestock, animals involved, of course, safety of the environment, can also cover intellectual property issues, benefit sharing, that it comes up as part of the ethical consideration. We always insist on, in factor intellectual property and benefit sharing, which are more society concerns, but you don't take and benefit from something intellectual property that's owned. It could be that could have been owned by some communities. So all those benefit sharing systems can be factored into regulation. The second general aspect of how we can promote ethical considerations is through what scientists have done for a long time, which is self-regulation by the research scientists themselves, whether they're in public or private institutions. And these self-regulations can be at institutional level, where the institution establishes a set of guidelines, but it can also be at individual scientific level. You must always operate with that principle that you should not cause greater harm to society. You should aim to protect society and advance humanity, but you cannot advance humanity without the environment, or environment being protected in the process. So the self-regulations can also be through professional. Under self-regulation, you can also have professional scientific associations like academies of sciences working together to ensure that the scientists who operate under their domain are working in a certain way. We can also have industry-specific associations. We have also seen Geoffrey talked about IGEM, which we will call an ad hoc international community of collaborators that also helps you, not necessarily industry association or professional or institutional, but it's an ad hoc community of groups of people that can actually keep exchanging ideas and saying this is beyond, this is not beyond, have you considered risks A, B, C, D? So that is still part of general scientific self-regulation. And then, of course, another way of self-regulation is through the scientific peer review system, as you noted from Hillson's discussion. Sometimes when you share your work or your proposed studies with the other scientists globally, they will help you understand things you may have missed, or because all these domains of ethics and safety. So when sometimes it's good to share with others, and then they give you feedback, and that is still part of self-regulation. So peer review helps support scientific rigor in our work, so that we ensure that whatever we do gives the society the greatest benefit at the lowest risk. And of course, self-regulation can indeed complement government regulation, and many times use hand in hand. Where there is government regulation, if there's no government regulation, we have seen self-regulation also work in many cases. And of course, it allows scientists to promote ethics and scientific considerations across the research and development pipeline. From the day you have your ideas conceived to the time you're rolling out a product to the community or to society, across the board, you must always ensure that you take care of ethics or safety issues along the way to make sure that the public is convinced and happy with your output, and would be glad to welcome your output. Yeah, we have also seen, of course, institutional policies that we have been developed by some institutions and implemented through institutional research ethics committees. We have also seen institutional research review boards. Yeah, I'm happy here. Nathan talked about this as well. Some agencies don't have this. Some agencies lump all other research efforts that they have within a university or a private sector company or a research agency into one institutional review research board or something like that, or ethics review committee. that will understand everything, every ethical question. Others do have separate ethical committees or research committees within their, depending on the discipline that they have. Others, an institution may create a synthetic biology ethics review committee, or use an existing institutional biosafety committee or an institutional ethics committee, as long as the expertise is there to support ethical review and consideration before as the research processes undertaken by the scientists. And we've seen that once the scientists do follow and document all their key ethical principles, the society is always comfortable with the products that come, as if these are documented, because you tell people all the considerations you have made, and through your risk assessment reports that will be generated during the research and submitted to regulation, even where there's regulation, people tend to be comfortable if you say you consider those issues along the way. And of course, there are many principles that scientists always have to think about. The benefit I talked about to society. You must not be heavily conflicted, as you do the work. You must have informed consent if you are going to deal with, say, other humans in some experiments. If you're going to, for example, have human subjects in your research, you have to have integrity. People have to, that has to be demonstrable, and many others, including privacy and confidentiality, professional competence. I'm happy Nathan brought up a matter of a desperate individual who really wanted to help his family member, but did not have the competence in the field and had to be referred to other people, because that is all part of ethical considerations. You cannot just say, I want to do it because you are extremely desperate. At least get the professionals to start working with and make sure that they do things in a disciplined manner, in systematic and disciplined manner. Of course, self-regulation can also be in form of an institutional moratorium, which we've heard about, but not very, very common. But some institutions say we'll hold on with this, we'll stop at this stage, we'll not proceed beyond this, this age of the embryo. All those are some kind of moratorium that help institutions also build confidence in society. Yes, but I would definitely recommend that institutions should definitely work on policies, and those policies should support the government regulation where they're there, and when they're not there, at least they will support the scientists to do work that will be acceptable. The next area that I would think is the third area, which I would consider is essential for us to promote ethical consideration, is what I would term in education, awareness, and outreach. And we must, any ethical consideration in research and development activities will not work if the researcher doesn't understand the ethics issues involved. If he doesn't fully understand, he can either be complemented through education, if it's necessary, or awareness is he or she is brought to light and is told what it's about and why they should be considerate of ethical issues, but also through sustained outreach. So the education can be early and continuous, early in the sense that at university or high school level, wherever you are being taught biological sciences and experimentation, they bring these concepts to your mind and explain to you what it means to be working with ethical principles, because you should aim at benefiting society with the least risks. So that should start, because experimentation starts at an early age, and people should be taught that you should not create harm. Of course, we encourage people, unless you're going for the military research, which is normally not taught at a young age, should always aim for minimal harm to society. So, and this can be implemented through direct curricular review and implementation of that curriculum within the universities or colleges where biological sciences are executed. And of course, we should focus those discussions or this education to the risks from unethical use of biosciences, and particularly the potential for the potential that we know for biological products or living things to reproduce or multiply, and that may perpetuate forever or if unchecked. So because biology is not, unlike a phone that you'll make and will be used for a time and will die or will be thrown away, a living thing can most of the time replicate and grow or reproduce in the environment. So if you made a mistake and you did not follow proper ethical considerations and produce a product that may harm the environment, it's likely to continue growing the environment if it has any fitness qualities attached to it, and that may harm society continuously. So we have to bring that education early enough in our discussions with our young people when we're training them. And of course, then the second component is the continuous, or the second component, which I will call education and outreach. And awareness. The second one is continuous awareness and outreach among the key stakeholders, who can include the potential users, that's bring the communities and society on board. And this, you can also reach out to make the regulators aware of the ethical issues involved, and so that they factor into their discussions, bring policymakers to get on board, like we had about when Geoffrey was discussing. We must involve leaders and tell them what we need to do to have the right policies, whether you're funding or you are absolutely just going to regulate or providing, what we call in some African countries, just policy, just guidance on how to utilize the technology. That can also factor in ethical consideration. But if you can't reach out to policymakers, you will not achieve that. So we can also do outreach to product developers, the scientists themselves. We still have to reach out to them. Much as we will have had education programs for them, we must continuously reach out and explain the new domains or the emerging domains of the field and its implications for society. Yeah, and research and mentorship programs, if any, and as well as I had some people in the text requesting for mentorship programs. Those mentorship programs must also contain awareness about ethics issues as you're planning to do any work in synthetic biology, so that we build a natural culture of ethical research in R&D generally, which of course in this case we're emphasizing synthetic biology. So in conclusion, I would say our lessons that we have from applications of modern biotechnology in agriculture and human health and animal health have shown that the transparency about ethics and safety issues generally increases society confidence in products, and therefore we must maintain that transparency, and we cannot maintain that transparency without documenting what we are transparent about, what we are doing, how we are doing it, and what the product looks like, and what the product does, and what the potential risks from the product, the benefits, again, potential risks from that product. Government or self-regulation is the best way to get the public on board naturally, of course, as we educate them or reach out to them through proper awareness and outreach programs. And always remember, I think we should always remember that it's about the benefits versus the negative risks, not just risks. I'll stop there. Thank you very much.
All right, thank you so much, Mr. Herbert. Thank you so much for that. Also very enlightening presentation. Thank you for shedding light on the importance of ethical, setting the right ethics to guide biosciences. From your presentation, I was excited that you actually highlighted so many points, which include policies, setting the right policies, the right guidelines. You also, I mentioned, which is very, very important, you mentioned the role of educating the researchers, educating the public, because a lot of people are not yet aware. They don't know what ethics actually involve. They don't know the boundaries and the borders they should operate their innovations. You mentioned that it's very important that we educate, we organize outreaches to educate people, because before you start penalizing anyone, it's good to first educate them. And then you also did mention mentorship, the role of mentorship, which is very, very pivotal as well. Mentorship is so, so important. And thank you so much, Mr. Herbert, for taking your time to expatiate so much on how we can promote ethical consideration for modern bioscience, for synthetic biology as well. It's been a very rich conversation, I can say that. And I can see in the chat box that a lot of people have been asking, are they going to be, would they have access to the recordings of this webinar? I want to tell you yes, so yes. The answer is yes. So you're going to have access to everybody that has registered. We're going to be sending you the link on how you can access the recordings of today's webinar. And then also, I want to also commend everybody that has joined us. I can see we have somebody, we have Albert from Uganda. We did also have Albert from Uganda as another, yeah, we have Leko Albert from Uganda. We have somebody from Madina from Pakistan. Thank you so much for joining us. We did have Asom Miriam Sani Lawa with the Nigerian Center for Disease Control, but she's joining from Japan. Thank you so much for joining us. And I can also see in the chat box, a lot of people are asking, would they have access to how is the certificate going to go? Yes, at the end of our webinar, we do offer all of our registered participants certificate of attendance. That is, if you join the webinar, you definitely have access to getting your certificate of attendance. So let's just quickly run through the question and answer. I want to appreciate Dr. Nathan and also our speakers. I can see that a lot of questions have been answered in the chat box. Thank you so much for doing that. That will actually save us the time. But so much we want to appreciate everyone that has joined us. We want to appreciate our speakers, Mr. Geoffrey, thank you so much for joining us. Dr. Nathan, thank you for joining us as well. Mr. Herbert, thank you for joining us. And I must also appreciate the communication officer at the Symbio Africa, Mr. Perez Wayne. Thank you for joining us. We've been writing this together, and thank you so much for making today a successful webinar. So let's quickly just take through the questions that have not been answered. Somebody said, greetings, are there African schools that have synthetic biology related courses yet? That's the question that we have there. I don't know who wants to answer that. Mr. Geoffrey, do you want to answer that? Someone is asking if there are schools, African schools. I want to assume that it's African University. If their courses on symbiotic, synthetic biology related courses. Mr. Geoffrey, are you there?
Yes, I'm here. Can you hear me?
Yes, I can hear you very well.
Yes, sorry, my PC got trouble, so I'm now joining using my phone. Yes, thank you for that. Thank you for that question. And I think towards the end of my presentation, I mentioned that it is one of the challenges that we still have. There are no courses yet for synthetic biology in all the African institutions. So it is something that we are trying to work on. Last year, I happened to get a chance to go and take this advanced course in synthetic biology. This course, which it takes place at Cold Spring Harbor Laboratory in New York. So I also use that opportunity to look at the future of having such a course in Africa. I talked with the director of that program, and yeah, we are still in the process of seeing how we can bring such a course as a starting point of having a synthetic biology course in some of the African institutions. So we still don't have, but I'm working on that. Meanwhile, the government side or the public university side, they should also be able to work on that. At Makerere University here, there is also some progress. They want to have a course, a postgraduate course in synthetic biology, but probably when they are going to review the curriculum, they are going to include, but it's still a process. So at the moment, I can say we don't have any course in synthetic biology.
All right, thank you so much for that. It's also another question that I'm directed to you as well. Somebody is asking, do you offer internship in your organization? The person said, aside from conferences, does Symbio Africa offers internship for academics and students who are interested in pioneering this field in their own schools?
Yes, so I can say we at least we have been able to have one internship program, because we were running a program on pandemic preparedness and awareness throughout Africa. So we had to get an intern to work on that. So that is already a starting point of us having internship program at Symbio Africa. And also, if there is any project like the one I've talked about, and any student is interested to be part of that project, we shall always make an announcement and get a suitable candidate for that project. And also, as Symbio Africa, I've been in talk with different collaborators of ours, like from Northwestern University. We want to initiate an exchange program where we can send our students, even including our lecturers from the African perspective, to go and learn new techniques that are being done from the Western world, and so that they can transfer the new knowledge they've learned back home here. So it's also something in progress. We are in touch with Stanford University and Northwestern University, but probably before this year ends, we shall be able to send one a scientist from Africa to US to gain that experience.
All right, thank you so much. There seems to be a lot of questions directed to people wanting to work with you. I would like to just tell Mr. Paris, please kindly drop the email or the contact details of Simba Box. A lot of people are actually interested in working with Simba Africa. I have questions there. Do you have opportunities for graduate trainings? Can individual join you? Please do drop your social media platform link in the chat box for everybody to know, for all of our audience to be able to reach you, and then so that they can also ask, continue the conversation on how they can engage with the amazing work Simba Africa is doing and on the planet, on the African planet. I also saw that a lot of questions have been answered by Dr. Nathan in the chat box. Some of the questions include people pointing at the potential risks of synthetic biology, and I love the fact that most of the questions has been answered. Just to reiterate, okay, yes, a lot of questions have been answered, so I'm really excited about that. Just to reiterate, everybody joining this webinar, you're going to be having access to the recording of this webinar, and we'll gradually end of the webinar. It's been a very rich conversation, and even from our chat box, I can see many of our audience saying thank you to our presenters, thank you to our speakers, thank you so much. And we do say thank you once again for joining us. But it's also very important that I also let you know before we go today, that I let you know about the amazing work the amazing stuff that is happening in my organization. I want to tell you, just like what Mr. Geoffrey mentioned, that Simba Africa is going to be having a conference in July. Like I said, I'm glad that Mr. Perez already dropped the link to the Twitter page, and I'm sure he's going to drop more links on how you can access that. I also want to share something briefly with us, and that is we're going to be saving the date for the 9th edition of GET's annual conference on One Health about security. And the topic we're going to be having for this year is so amazing. It's quite very similar to another topic that we're going to be having, that Simba is going to be also having. And here is it. You can just look at your screen. We're having the 9th African Conference on One Health and Biosecurity. And this year's conference is focused on the theme is maximizing the benefits and understanding the risk of synthetic biology and other emerging biotechnologies in Africa. I can see that you've had an amazing section on synthetic biology. The conference is going to be focusing on it elaborately. We're going to be looking at the innovative parts. We're going to be looking at the research part. We're going to be looking at the data analysis, and we're going to be bringing in many speakers, both international, many of our experts, both local and international experts will be attending. And I want you to save the dates. The date is from the 1st to the 3rd November 2023, and it's going to be happening right in the city of Lagos, Nigeria, at Civic Center of Victoria Land. If you've been part of our conference in the past, this is the 8th edition. GET has been organizing this conference for the past 9 years, and I can tell you without missing any word, this is the biggest biosecurity conference in Africa, and you don't want to miss it. I'm also sure that our communication team will be dropping details of our social media platform, of GET social media platform, for you to get to know, because we're going to be releasing many updates about the conference, how you can be part of the conference, how you can register for the conference, how you can start preparing for the conference, the speakers that are going to be joining us for this conference, and also the amazing highlights this conference is going to be featuring. Once again, I'm glad that you joined us. I don't know what we usually do again is that for all of our webinars like this, somebody said, how can we get certificate? Yes, you can get your certificate. The certificate will be sent to you alongside with the recording. So if you have registered, I wanted to know that you're going to have a certificate. So I'm still expecting our communication team to drop details of this year's conference, of this year, the 9th edition of our annual conference of One Health Security, on our chat box for us to be able to keep up with some of the updates we'll be releasing in the later weeks on the conference. All right, I don't know if any of our audience have, well, you know, we always, when we have time like this, we just have about, let's say we have less than 8 minutes more, but I just don't want to end just straight like that. If there's any of our participants that will have something to say, I can definitely give you opportunity to speak. All you have to do is to raise up your hands if you have anyone that's interested. If you want to say something or you want to make a comment about today's topic, you can just raise up your hands while I give you opportunity to talk. Do we have anybody? Esteban that is interested? Ask me very fast because of our time. Anybody interested? I'm looking at the participant list to check if there is anybody that wants to raise up their hands. Okay, so let me see if there's anyone. All right, is there anyone? Okay, yes, that's fine. I'm sure a lot of people are enjoying themselves. At this point, we'll be joining the cutting of today's webinar. Like I said, guys, organized my organization, Global Emerging Pathogens Treatment Consortium. We normally organize a monthly webinar series. And thank you so much for joining us for the March edition. I want you to look out for the April edition, which is going to be amazing as usual. I want to appreciate our partner organization that we co-organize this webinar together, Bio Africa. Thank you so much to our dear speakers, Mr. Geoffrey. Thank you so much, Dr. Nathan Hillson. Thank you for staying up at early hours of today to, to share your expertise and your rich knowledge on this topic. And I want to also appreciate Mr. Herbert Oloka. Thank you so much for also joining us. We do appreciate. Thank you to all of our participants. Staying tuned with us, and we hope that these conversations can also continue. My name is Ifeoluwa Alabi. I am super excited to be your moderator for today, and I want to say for now. Yeah, I know bye is always very hard, but we definitely need to say bye now. Bye, everybody. Do have a wonderful rest of your day. Bye.
Bye.